Good evening, guys. I've got E for you, which is extra tonight for shows. We're talking about the effects on the brain of alcohol use disorder and, neurodegeneration and dementia, how it links to dementia. So, we'll go through that tonight for this show, which falls under brain health for me. Okay, certification brain health. So, with that being said, let's get going, shall we? Okay, alcohol use disorder produces a specific microglia driven immune signature in the brain that is linked to inflammation, neuronal damage, and features of neurodegeneration. Key findings: distinct neuroimmune state. Chronic alcohol use doesn't just cause general inflammation. Instead, it reshapes the brain's immune environment into a unique disease-like state that is different from the immune responses seen in Alzheimer's or other neurodegenerative conditions. Microglia at the center. Microglia, the brain's resident immune sentinels, shift from their normal surveillance role into a reactive, potentially damaging phenotype, their altered shape, protein expression, and transcriptional activity mark a profound change in how the brain responds to alcohol exposure. What the researchers did. Human post-mortem analysis. The study examined brain tissue from individuals with alcohol use disorder compared to age-matched moderate drinkers. Focus region: the orbital frontal cortex, a hub for decision making, impulse control, and social behavior, was chosen because dysfunction here is strongly linked to the behavioral symptoms of AUD. Multimodal approach. Researchers combined 3 layers of evidence: cell morphology, protein markers, and RNA transcriptional profiling to build a comprehensive picture of immune changes. Microglia and molecular changes. Morphology: microglia in AUD brains were visibly more reactive with enlarged cell bodies and retracted processes, classic signs of activation. Protein markers. Immune related proteins were expressed at altered levels, suggesting heightened inflammatory signaling. Transcriptional signature. RNA sequencing revealed coordinated changes in gene expression, pointing to a rewind immune program that links alcohol exposure to stress pathways and neural vulnerability. Links to neurodegeneration, pathways to damage. This immune signature is tied to mechanisms that promote neural dysfunction and eventual cell loss, offering a give me 1 second, guys. Offering a biological explanation for the cognitive decline often seen in long-term AUD. Distinct profile. Importantly, the immune response in AUD is not identical to Alzheimer's or Parkinson's. It represents a specific trajectory of neuroimmune disruption. Through it, though it may intersect or exacerbate other neurodegenerative processes. Implications and next steps. Clinical relevance. Identifying this immune state opens the door to therapies that specifically target microglia activation or the signaling pathways involved, potentially reducing alcohol-related brain injury. Future research. Key questions remain. Are these immune changes reversible with abstinence? Do they predict who will develop severe cognitive decline? How do genetic risk factors or co-occurring conditions like depression or liver disease shape the responses? Broader impact. The work reframes alcohol use disorder not just a behavioral or social condition, but as a biological disease of the brain's immune system, with implications for treatment stigma, and prevention. Would you like me to map out how this AUD immune signature compares to the microglia profiles in Alzheimer's and Parkinson's disease? So you can see where the overlaps and uh, diverse things lie that could highlight whether AUD accelerates or mimics aspects of classical neurodegeneration. Okay, guys, so that is it for your shows tonight. I will be on tomorrow for shows, or later. Till then, Gator Wellness, take care, folks.